Hi, everybody. We are back and getting ready to record some more of these small mini lectures to help you keep up with the textbook. This is actually part two of the introduction, so these slides are originally found in the lecture slide one in the lecture slides portion of both Blackboard and on Piazza. So let's go right ahead and take a peek at our initial idea. Here we have the concepts of the basic principles of genetics. This is a classic little cartoon from a science cartoonist at xkcd.com. They're usually pretty funny. And this really really outlines some of the things we'll be talking about chromosome structure, function, and of course, meiosis. So we're really in good shape. A quick reminder that if you'd like to focus in on one of the slides, either to get a screenshot or to take some notes or whatever you'd like to do, remember there's always the pause button so you have an opportunity to listen up. What we'd really like to do now is remind you about the structure and function of DNA. We will talk more about the chemistry underlying this, but notably we have eukaryotes, and we're going to skip over prokaryotes for a moment. We'll discuss prokaryotic genetics in the lab in a couple of, probably in about 2 weeks. Notice that we have the molecule of DNA. It is a double helix. In eukaryotes, but not prokaryotes, the nucleosomes wrap the DNA tightly around them. There are 8 histone molecules that are clipped with histone protein H1. And of course, we become coiled, more coiled, super coiled, eventually till we get to the size of the chromosome. Now, this really is an overview for you and a reminder that you really need to review either your Bio 1 notes, your Bio 1 textbook, and I have a link a little bit later on that'll give you some idea of where to go if the genetics text is a little too complex for you. Basically at the OpenStax library, where you can get a free biology textbook. I'm going to zoom through a couple of slides because these are things that you should already be familiar with, but important that you understand the chromosome vocabulary. What is chromatin? That is the threads themselves of DNA, euchromatin, heterochromatin. You should also be familiar with the idea of ploidy, that is the nature of the number of chromosomes that organisms have. You should also be familiar with the concept of the chromosome. One thing that students have a little bit of trouble with, in fact, even professors have a little bit of trouble, is that we use the term chromosome for situations where a chromosome contains only a single molecule of DNA, and this, of course, is known as a single chromatid. But we also call a chromosome the situation where we have 2 sister chromatids. And so you see this is actually 2 independent molecules of DNA that are connected at a structure known as the centromere. So you have to be a little bit careful. If I were king of the world, I would call one a monochromosome and the other one a duochromosome or a diploid chromosome, some other name to differentiate. But of course, nobody called me at this point in time. Notice that depending on the position of the centromere itself, the chromosome can take on a very, very different look when we look at actual chromosomes. Here, of course, is a karyotype. That is, the chromosomes are stained at metaphase of meiosis. If you don't know what metaphase is, of course, you really need to go back and review your Biology 1 textbook. but also there's an excellent discussion in Pierce as well. And notice that the position of the centromere in the chromosome actually is a classification method. If it's in the middle, it's metacentric. If it's slightly off the middle of the chromosome, it is submetacentric. Of course, acrocentric is near the end, and telocentric is where the centromere is actually located at or extremely close to the telomere of the chromosome. When we line up the chromosomes, we get a karyotype. And here is a karyotype of the human genome. Notice we have homologues. This is a position. Now notice we've got the 2 chromosomes, 2 molecules of DNA connected by the centromere, and these are homologues. So notice we've got them classified here, and by both size. So the very, very largest, or chromosome 1, is a metacentric. And notice we begin to get submetacentric. So primarily, chromosomes are classified by size. The biggest, of course, is chromosome 1, down to the smallest, which is chromosome 22. And then separated by, after size, also separated on the basis of the position of the centromere, and you'll notice that 20, 21, and 22 are all telocentric. As you know, there are 23 pairs of chromosomes in human beings, and notice that we have a position here. Here is the 23rd, or the sex chromosome, which of course in humans and in all of the mammalian species is the X chromosome. 2 X chromosomes, of course, means that that karyotype was taken from a female. And I don't want to give the impression of 2 things. First of all, not all species have the same total number of chromosomes, and not all species determine sex on the basis of having 2 X chromosomes. There are other ways, and we'll briefly review them. First of all, to give you a sense of the tremendous variation in chromosome number, notice that the simplest of the eukaryotes, yeast, mold, algae, have anywhere between 7 and 17 different chromosomes. Maize, which you should be reviewing or will be reviewing this week in lab, has 10 chromosomes. Notice our buddy the fruit fly has 4, mosquito has 3, and of course humans, or Homo sapiens, and our closest relative or related species, the chimpanzee, has 23 and 24 chromosomes respectively. I happen to like, one of my favorites here is of course the chicken, which has 39 total chromosomes, really quite a few. Now, how, of course, is sex determined? Now, I noted that XX, meaning 2 X chromosomes in the mammalian species, dictates a female. However, it is actually the Y chromosome that dictates and determines the sex of the offspring. The Y chromosome carries the SRY gene, which produces the protein, the testis-determining factor. This gene is expressed at the end of approximately the 3rd trimester of a pregnancy or of embryonic development, and at that point in time it shifts the development of the embryo towards the male. But there's some interesting potential defects. We can have the normal male, and again we use that word normal very judiciously. Normal, of course, is only a setting on your washing machine and not something we should use when we talk about biology. But a typical male has, of course, the Y chromosome plus the testis-determining factor. Of course, there are several genetic conditions, one in which the SRY gene actually gets translocated to the X chromosome, so you could actually have an individual with two X chromosomes, but because of the location of the SRY gene, actually becomes an XX male. Alternatively, we know of several mutations in the SRY gene that cause testis-determining factor to be absent or malfunctioning, which means that the embryo then develops completely along the female tract. So kind of an interesting element to sex determination. When we think about the ways that other species can determine sexes, our buddy Drosophila has an interesting perspective, and the determination of sex has to do with the ratio of X chromosomes. Now, in Drosophila, I mentioned earlier that Drosophila has 4 chromosomes. They are actually numbered 1, 2, 3, and 4, and it is the number 1 chromosome that is actually considered to be the X chromosome. But it's a little bit of a misnomer because of the historical interpretation of sex determination and not related to our current understanding. But notice that you can have a ratio of X chromosomes to autosomes in which the ratio dictates whether the organism is phenotypically male or female. So there are really some kind of unique combinations of chromosomes found in Drosophila. Here's one of my favorites. This is a recent publication from 2018, and this is the sex determination system of the Smoky jungle frog. And the interesting thing about the Smoky jungle frog, and you can see the karyotype here, in this case, the diploid number of chromosomes of the Smoky jungle frog is 22, and in fact, more than half of its total number of chromosomes are actually sex chromosomes. So 12 out of 22 are actually sex chromosomes, and you can see them numbered both X's and Y's. This beats, of course, the previous champion, which was the platypus, which has a diploid chromosome number of 52, but only, sadly, 10 sex chromosomes. So the platypus, which used to end up in all sorts of genetics textbooks, was displaced now in the new books by the Smoky jungle frog. Of course, the other method is what's called the ZW system. And in this case, this is typically found in birds, but also in butterflies. And in this case, the ZW genotype is female and the ZZ is male. So in this case, what this means is, unlike in the mammalian species, where the male determines the sex of the offspring, that is by providing the Y chromosome, in birds, for example, the female determines the sex of the offspring by the contribution of either her Z or W chromosome. So it's a little bit different. Curiously, we do not know the gene located on either the Z or the W, or the protein that actually causes the change in development into a particular sex. So we really do not understand completely sex determination in this other chromosomal mechanism of sex determination. There are a couple of others predominantly found in amphibians, but also in certain reptiles. This is the Haplo-Diplo system, and this is where a diploid individual is female, a haploid is male. And in this case, notice we have a situation where an unfertilized egg actually develops into a haploid male. The haploid male of course, is created the process of an unfertilized egg developing into a complete organism is called parthenogenesis. So we've got an interesting situation here. This particular system is often found in insects, and in particular the social insects. A final type we'll chat about here is, of course, temperature-dependent sex determination. This is found mostly in reptiles, and what's really kind of curious about it is there are two different patterns of temperature dependency. The first to be observed was in turtles, where low temperatures increased the percentage of eggs that hatched as males. High temperature indicates that there is a low percentage of males, and so this is called pattern 1. Alligators have kind of a curious one in which either of the extreme temperatures results in very low number of males. Moderate temperatures indicate a very high percentage of males. More than likely, this has to do with the environmental conditions that cause an increase or decrease in food resources for the organisms. So you could see there's a tremendous interaction between the environment and sex determination in reptiles. If you are a little bit confused, because we're going to shift gears a little bit, and if you do not recall all of the stages of meiosis, there's an excellent discussion again in Pierce. If it's a little bit complicated for you and you need to revisit your Bio1 information, please do so. And in case you sold your Bio1 textbook, notably there is a free online textbook located at openstack.org, and the genetics section, there's a single chapter overlying genetics. It is the second edition of the biology text and is actually a very, very nice introduction, not too complex, and will give you a great jumping off point to then read the chapters in Pierce. I'm not going to spend a tremendous amount of time right now on this, but a quick reminder that during meiosis, crossing over takes place, and this is where the homologous pairs of chromosomes line up. They are connected by a series of proteins, a chiasma forms, in which case the genetic information on one portion of the chromosome gets separated and translocated to the other chromosome, and defects in this process can lead to all sorts of variation in chromosomal number. Notably, in older women, the failure of this complex to separate can cause genetic disease, and it is the failure of the two chromatids during this position to separate completely. So instead of getting, as in anaphase and metaphase here, instead of getting 4 independent gametes, you'll get one particular oocyte that does not separate, so you get 2 chromosomes, and in this case, chromosome 21, in the developing organism. And of course, it looks something like this, a little bit easier to see. We see in meiosis 1, the reductional division, and then it is in meiosis 2, where we see that these 2 sister chromatids do not separate. So you basically get one organism with none of that particular chromosome and 2 of the other, which means that you end up, when these 2 oocytes, if they are both viable, are fertilized, they become trisomic or monosomic, as opposed to the normal diploid number of chromosomes. Of course, this results in Down syndrome when it incorporates a triploid condition in chromosome 21. It is called Down syndrome, not Downs syndrome, because it was first identified by John Langdon Down. It was not identified as a chromosomal abnormality until 1959. So very interesting and fairly common rather, mutation that occurs in humans. But there are others, and we can see it is not the only trisomy of an autosome. There is another one, which is trisomy 18, in which case we see a trisomy that is 3 chromosomes for chromosome number 18, has very, very similar phenotypic expression as Down syndrome or trisomy 21, but is not nearly as common. There is also a typical XXY individual. This is known as Klinefelter syndrome and tends to cause a male because, remember, this results in a male phenotype because of the expression of the gene SRY in Testis-Determining Factor. However, because of the additional X chromosome, the individuals that are Klinefelters tend to be feminized. That is, they have some female characteristics as well. There is also the triple X individual. This is very common, sometimes just known as XXX or 3X or triple X. It's described in a variety of ways. So these are individuals, they are female. They tend to have some reproductive difficulties because of the inability of their germ cells to undergo meiosis properly. There's also a situation where we have an individual that has a single X chromosome and missing a Y chromosome. So this is an aneuploidy with the prefix an, meaning missing or deleted, and this individual is just an XO individual, and this is known as Turner's syndrome. And Turner's syndrome, again, tend to have some endocrine abnormalities, difficulty undergoing meiosis, and you can really, but very, very little of the cognitive difficulties that are so often seen in other genetic abnormalities. One that's been very controversial over the years is what used to be called a supermale because it has 2 Y chromosomes, but is now just known as an XYY individual. And it was called a supermale individual because biologically it was thought that the extra Y chromosome caused a spike in testosterone levels in the males, and of course, in the 60s was thought to be associated with an increase in criminality or criminal behavior because they went through the prisons and karyotyped individuals and found a high percentage of XYY individuals, or at least higher than they thought. However, they failed to compare them to the incidence of XYY in the general population, and it turns out there is no such biological relationship to the XYY and increase in testosterone, nor is there any relationship to criminality. So it's an interesting perspective on chromosomal information. So this took a little bit longer than I thought, but I wanted to get through looking at some of these different karyotypes. Catch up, please, in Pierce, and we'll be moving on to the basic rules of inheritance in the next little mini lecture. Take care, you guys. Be safe.